line of an ancient sea, the ocean strikes both fear and wonder into the hearts of men. Fear because the ocean can be hostile and unforgiving. Wonder because although we harvest its bounty with pots, nets, and lines, many mysteries concerning the inhabitants of the deep remain unsolved. This film tells of a visit to the ocean bottom near Kodiak, Alaska. The visit resolved a fundamental question about tanner crab biology, but like most explorations, the voyage raised more questions than it answered.
In the winter when it's drab, I'll go fishing for some crab. Fishing, crab fishing.
The tanner crab, Chionocetes bairdi, and its cousin, the snow crab, Chionocetes opelio, collectively support commercial fisheries in Alaska worth over $200 million annually. Crab abundance fluctuates widely. When crabs are plentiful, fishermen do well.
And every now and then I'll pull another in.
The reasons for booms and busts in crab populations are poorly understood. Among factors influencing crab abundance are ocean temperature, wind and current patterns, disease, predation by finfish, food abundance, and fishing pressure. Of these, only fishing pressure is directly controllable by man through management. Rational fishery management depends on knowledge of life history, behavior, growth rate, and abundance. In Alaska, the National Marine Fisheries Service and Alaska Department of Fish and Game cooperate to perform research on crab biology and to manage the crab fisheries. Management of tanner crab fisheries is accomplished by allowing fishermen to harvest only a given proportion of male crabs larger than 5.5 inches. The implicit goal is to make sure that enough males survive the fishery to mate with every mature female. Laboratory studies have shown that a single male bairdi can fertilize around a half-dozen females. Studies have also shown that a female can retain spermatozoa from one mating to fertilize several clutches of eggs in consecutive years. Male tanner and snow crabs undergo one molt, during which they acquire a large claw. Biologists have always assumed that this measurable increase in claw width indicates that the male crab has reached sexual maturity and is capable of successfully mating in the wild. This assumption is key to the determination of a minimum legal size, yet it had never been tested prior to 1991 by observations in the ocean below scuba depth. The question is complicated by the fact that small claw males do have mature gonads and can mate with small females in laboratory aquaria to produce fertilized eggs. Thus, there is a difference between gonadal maturity, the ability to produce viable spermatozoa, morphological maturity, the possession of a proportionally large claw, and functional maturity, the ability to mate with mature females in nature. In the autumn of 1990, Dr. Bradley Stevens of the National Marine Fisheries Service Laboratory in Kodiak, Alaska, was pondering the knowns and unknowns of tanner crab reproduction. He decided to examine the assumption that morphological maturity is equivalent to functional maturity in bairdi crab. He and other biologists had attempted to study mating pairs in shallow water using scuba, but had found only small animals. Of 36 mating pairs collected in Women's Bay on Kodiak, not one male had been larger than 4.5 inches.
So to get the larger animals, we have to go deep. And we can't use a trawl to capture them because when you do that and they come up, they're all jumbled up, and you don't know who was really mated with who. Likewise with pots. mating animals will not enter pots because they don't feed. If they did enter pots, they might end up mating with whoever happened to enter the pot rather than someone they would normally mate with. So the best way is to actually go there and find them in the act of mating and bring them up.
In order to go there, Dr. Stevens realized a submarine would be required. He submitted a research proposal to the NOAA-sponsored National Undersea Research Program. It was approved and funded by the West Coast Undersea Research Center at the University of Alaska in Fairbanks. Research began in Chiniak Bay off Kodiak Island in mid-April 1991. The submersible which was selected for the job was the Delta, a 16-foot research submarine owned by Delta Oceanographic of Torrance, California. It can operate to a depth of 1,200 feet and carries a variety of instruments for sampling water, sediment, or animals. It carries an external video camera and 35mm camera, as well as searchlights and strobe lights. It also has a remote tracking system, so its position can be monitored by a 120-foot surface support vessel, the Pirateer. When underway, the pilot sits on a stool above the observer and looks out the portholes in the conning tower. A biologist observer lies on the floor and looks out the front portholes.
We set an initial goal of capturing 50 mating pairs, but this was just an arbitrary number, and I really had no idea whether it was realistic or not. As late as 1 month before the sub was supposed to arrive, I really had no idea how we were going to capture crabs, but I knew we'd find a way. We built several experimental traps, which all worked great in the lab, but they failed miserably underwater for one reason or another. When the last one failed, I flew down to LA to see the sub and find out what it could do. It had a mechanical arm with 2 fingers on it, and I thought with additional fingers it might make a great crab grabber. So the owners gave us the blueprints, and we had Bill Nippus, a local biologist and machinist, create additional fingers for us in the form of a hand.
The observer inside the sub operates the grabber like a pair of salad tongs, using a pair of handles to open or close it. It can be extended 3 feet from the sub and rotated 360 degrees. It was very effective at capturing boots as well as crabs. To launch the Delta, it is hoisted off the deck with a crane and lowered into the water. Then it is secured to the side of the Pirateer against a mat of tires. After the sub is secured, the divers enter it through the top hatch. First, the biologist climbs in and lies down on the floor. Then the pilot climbs in and secures the hatch. When all is ready, the deck crew releases the safety lines. The pilot starts the motor, and the sub cruises away from the ship. The Delta can travel at speeds up to 3 knots on the surface, which is necessary for quick maneuvering near the Pirateer. On the bottom, it usually travels at about 1 knot. When the sub is in position to dive, the pilot opens the top valves on the front and rear ballast tanks, allowing air to escape as water enters the tanks. Here you can see the air bubbling up out of the tanks as the Delta descends. The first day of the project was spent testing the sub and the crab grabber. Scuba divers assisted and photographed this activity in 45 feet of water. Divers placed crabs near the sub so that the biologists could practice catching them. Here comes the crab on the right side of the picture. The biologist reaches out and captures it in one smooth motion, then picks it up and places it in the basket. a task which is not as easy as it seems. With a little practice, coordination between the pilot and the biologist improved, and the operation became much smoother. After capturing the crab, the biologist withdraws the arm into the sub. The crab sits in its basket. The pilot prepares the sub for surfacing. And the Delta is finished with her test dives.
Now, when do they actually mate? When
This time of year.
this time of year is when they're actually mating.
Yeah, she'll hatch the eggs and the male will come over and grab her.
Oh, so right after those go, he's ready for...
Yeah. Sometimes they grab them before and they wait for the eggs to hatch. Now, she doesn't have to mate every year. They store sperm and she can fertilize an egg clutch for 2 or 3 years in a row.
Now, I thought I heard you say earlier something about the male carries the female around for a while. Or?
Yes. He'll grab her right there, see? That's how they make that mark.
Oh, I see.
We see them, they'll be running around on the bottom and hold the females like this, protecting them.
Running around, just hanging on to her till the right time.
Yeah, the right time.
Oh, be dark.
Despite the fact that tanner crabs have been harvested commercially for many years, we really knew very little about their reproductive behavior. When scuba diving, we know we can find small crabs mating in shallow water, scattered randomly about. We thought we'd find crabs scattered randomly about in deep water also. We knew from conversations with fishermen that crabs tended to concentrate in certain areas of the bay, and we planned to spend some time looking for these, but we thought they'd be rather large areas of the bay.
On the second day of diving, co-investigator Bill Donaldson of the Alaska Department of Fish and Game found an area in 500 feet of water where male crabs were concentrated up to 5 to 10 per square meter. This was a much higher density of crabs than expected, but no grasping pairs were seen.
Roger, roger, any crabs?
After a few more days of unsuccessful searching for grasping pairs elsewhere, the scientists decided to return to the male aggregation and explore it more closely.
Yeah, outside the one, yeah.
More crabs. More crabs. Getting into some big males.
Close now. Another big one on the left. That's the 1, 2, 3, 4, 5.
We're starting to see quite a few crabs.
Here's some females, I guess.
After seeing no crabs for a long time, they suddenly saw many female crabs right in front of the sub.
Yeah.
There's a big sculpin. There's a whole bunch of them there. There's a lot of them.
Oh, wow. Let's just drift here, and I'm going to try to get some pictures. Actually, if we could scoot forward a hair. Just a hair.
This is thicker than we put the thing in.
God, they're mounted up on top of each other.
Yeah.
I have too much equipment here.
The sub began to run into larger and larger piles of female crabs. Some of them were stacked 3 and 4 layers deep.
It was amazing. I never expected to see anything like it. At first there were no crabs, and then there were literally hundreds of them. They piled up against the sub so thick I couldn't see out the porthole. Crabs were falling off the side of the sub past the portholes, but the visibility was so poor I couldn't see more than a couple of feet. I thought this was a phenomenal discovery, but it was really just the tip of the iceberg.
Over the next few days, the scientists learned the true nature of their discovery.
We got a crab pile. There are several. They're much smaller pods of about 1 meter across. I see 4 of them, small piles of crabs.
Instead of one large pile of crabs, there were many small piles, each about 2 meters apart. Surrounding this central area of piles was a ring of males at densities of up to 10 per square meter, including occasional grasping pairs.
They're separated, what, 2 meters maybe?
Yeah.
Sometimes a little less. Oh, they're all over the place.
On one transit across the aggregation, 12 piles were counted. Some piles were low and elongated, and others were high and steep-sided. Each pile consisted entirely of mature female crabs, carrying eggs which would hatch into larvae within a few weeks, after which the females would mate again.
Keeping the same heading at 250 degrees?
Yes.
Okay. Absolutely.
Your course is very good. Your committed is very good.
Roger.
Approximately how many crabs are in your pile?
The piles varied from single-layer mats of 50 crabs to large mounds of over 500 crabs. The average pile had about 300 female crabs, was about 1 meter wide and half a meter high. Outside of the aggregation, there were very few crabs for miles around. The scientists eventually were able to estimate that the entire aggregation was about 150 meters long and 50 meters wide, just slightly larger than a football field.
Okay, there's one right here, right in front of me on the right.
Okay. On the right. So you go straight ahead. How far out on the right?
That's maybe up 1 meter.
Nice. The whole screen full of crabs.
The scientists guessed that there were about 200 piles of crabs present, using a conservative estimate of 250 crabs per pile and a sex ratio of 10 females per male. The entire aggregation was estimated to contain 50 to 60,000 crabs.
Hit them in about 30 seconds.
The winds are building up here. We might have to pick you up pretty quick.
Okay.
Roger that. We have this huge pile right in front of us. Really super.
With a crab in his mouth. God, what a picture. Is it on video?
Yep, got it on video. Got the fish.
We gotta get this camera working.
This unexpected discovery was so exciting to the scientists, they were tempted to spend the rest of their dive time examining the female piles. But the major goal of the project was to capture grasping pairs, so the scientists reluctantly concentrated on their crab grabbing.
I see a mating pair in the video screen straight ahead.
The biologists tried to catch both crabs of each grasping pair with the grabber, but often the female was dropped as the pair was being stuffed into the basket.
I've got a grasping pair on the right.
You do?
Yeah. Stop. And this one is male, is female, so I'll leave the male. I will get them both this time. Okay. Okay, you see him? He's right on top of the female. They're actually mating. Just off to our right. Okay. Okay. Spin, just spin. Okay, stop.
I doubt we missed a
Got him! Got him! In the act! But why doesn't interrupt us? Drop that basket.
The basket had 2 compartments which provided separate quarters for 2 pairs of crabs to avoid swapping of partners during the return to the surface. On the surface, length and width of the shell was measured for both males and females. On males, the size of the claw was measured, and on females, the width of the abdomen was measured. Grasping pairs which came up together were placed in tanks of seawater on board the privateer. Many of them began to release larvae.
Most of the crab that we caught weren't actually in the process of mating, they were just grasping. And we assumed from that that they would soon mate. In order to make sure that that assumption was correct, we held pairs in cages to see if they produced clutches. All the females held did in fact produce fertile clutches within a week or so after capture. We then transferred about a dozen females into an aquarium and monitored their egg development for about 2 months.
By the end of the project, the scientists had learned many new things about crab biology. The most exciting and unexpected of these was that the crabs formed into a highly structured aggregation in a very small area of the bay in order to reproduce. But they also accomplished all of their original goals and a few additional ones as well.
One of the goals of the project was to capture 50 male tanning crab grasping mature females. We were able to capture 47 males, and we actually retrieved 17 intact grasping pairs and brought them back to the surface for observation. Additionally, we captured a number of adult females off of the pods of females that we observed on the bottom, and also captured free roaming males and females. An additional objective was to help define what we mean by the term sexual maturity, and if a large clawed crab is a crab that mates with females. We do know that small male crabs with small claws can successfully fertilize a female, but the question is what size males actually mate with females in the ocean environment.
This graph shows the size of the male's claw in relation to the width of the crab. The lower cloud of points are all small clawed crabs, and the upper cloud are crabs with large claws. Many of these crabs were collected by trawling from a ship. All the male crabs that we captured grasping females, either by scuba diving or with the submersible, had large claws, except for a few that had regenerated claws. Out of 103 grasping males, only 1 was a true small clawed crab. This means that even though they are physiologically capable of mating, most males do not grasp and mate with females until they acquire a large claw.
Another thing we learned is that larger male crabs tend to select for larger female mates, up to a point such that the female is always about 75% of the size of the male. But since the females mature at about 4 inches and don't grow after that, the larger males have a much more limited size range of females to choose from. This year we focused almost strictly on learning about the male crabs mating with the old shell multiparous females. In 1992, we'd like to study more closely the behavior of the females. For instance, how many crabs are there in a pile? How many piles are there? How large an area does it cover? How long does it last? Why are some females in piles and others not? We've requested more submersible time for 1992, and we've also requested the use of a side-scan sonar, which can resolve objects on the seafloor as small as 6 inches. Hopefully using this, we could find an area of piles of crab much cheaper than with a submarine, and then bring the sub in to examine it more closely and collect samples from it.
During the course of the project, the biologists accidentally discovered aggregations of another crab species called the lyre crab, Hyas lyratus. Mature female lyre crabs are so small that the males can hold them up off bottom with one claw to keep them away from other male crabs. This species is very closely related to tanner and snow crabs, and it suggests that mating aggregations may be a characteristic of the family Magidae, which includes all three species.
Brad, what does this all mean for crab fishermen or fisheries managers?
Well, our research this year has given us new information on the reproduction of tanner crabs, and we now know that it is only the large-clawed males that participate in mating. And maybe we can use this information to alter our management strategies, perhaps by changing the legal size limit or setting a size limit based on claw size. Regarding the aggregation, that in itself has a number of implications. The primary one is it may be very susceptible to disruption by trawling, so perhaps we ought to be protecting the crabs during this period. But another major implication is in stock assessment. For those of us like myself who have to estimate the abundance of these crabs, we often come up with numbers where we have no crabs, no crabs, no crabs, and then all of a sudden we catch a lot of crabs. This phenomenon, this aggregation, may explain why we get these kinds of results, and they drive statisticians nuts because it's very difficult to use them. On the other hand, perhaps we can use this behavior to improve our estimates. If we can locate aggregations using some equipment like a side-scan sonar or an ROV, a little underwater camera, then perhaps we can target our sampling on the aggregation and increase the precision of our estimate. But there's another reason to be excited about this, and maybe that means more to the average person. It means that it's still possible in this day and age to visit a part of the planet that's seldom visited and discover something just totally new and unexpected. And it leads us to think about these animals in totally new and different ways. They communicate with each other, they segregate, and they cooperate with each other. But most of all, the discovery of this incredible, wondrous phenomenon is something I hope will open up the minds of all humans who see it to the endless possibilities that exist in nature.